Hello everyone. Welcome back to Dr. Liu's VCE Biological channel. Today, let's learn together about the 2020 VCE Biology. We will focus on questions about the regulation of biochemical pathways. My explanations may need to be revised. If any teachers, parents, or students find errors, please feel free to provide your guidance. Since the diagram is not provided, we cannot analyze questions 1, 2, and 3. There are 3 test tubes. Each test tube contains the same enzyme, but different substrates. Test tube 1 contains substrate A, test tube 2 substrate B, and test tube 3 contains both substrate A and B. In test tube 2, enzyme plus substrate B does not produce a new molecule and return to the initial states. So, substrate B could bind with the enzyme as a reversible inhibitor. When the substrate B is mixed with substrate A, substrate B will compete with the enzyme with substrate A and decrease the concentration of product in the final contents of test tube 3. The primary structure of a protein determines the higher level of structures. This diagram shows the relative activity of an enzyme versus temperature. The highest relative activity is at 57 degrees, so this enzyme's optimum temperature is around 57 degrees. This question is about error bars. The smaller the error bar, the more accurate. The error bar of 52 degrees Celsius was smaller than that of 46 degrees Celsius. So, the measurement of 52 degrees Celsius was more precise than that of 46 degrees Celsius. Adrenaline is an amino acid-based hormone. The image below shows a cell section of an adrenaline-secreting cell examined under a transmission electron microscope. A secretory granule is a large vesicle formed when several smaller vesicles fuse. Each secretory granule contains a large amount of adrenaline, which is stored until the cell receives a signal to release it. Some organelles in this photo are labeled as GA and M visible. GA is the Golgi apparatus. One of its functions is to modify the protein into adrenaline or package adrenaline into vesicles. M visible is mitochondria. One of its functions is to provide energy for the synthesis of adrenaline. Exocytosis is a cellular process where a cell transports molecules out of the cell by releasing them from vesicles that fuse with the cell membrane. This active transport mechanism is crucial for releasing neurotransmitters, hormones, and waste products. Secretory granules containing the adrenaline fuse with the plasma membrane and release their contents. Mitochondria produces energy. Some students wrote very long and detailed descriptions of the complete process of synthesis and concluded with the correct answer. While the description of synthesis was irrelevant, but not incorrect, marks were only awarded for the correct answer. Students don't need to use valuable time to write irrelevant answers. In the regulation of biochemical pathways, 7 multiple-choice questions are worth 7 marks, and 1 short-answer question is worth 5 marks. The overall score for this section is 12 marks. With a total possible score of 120 marks, the percentage for this section is 10% of the total score. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.